Hello, interstellar everyone. Welcome to class 11 biology. In this video, we are going to discuss about the first phase of meiosis, that is prophase 1 of meiosis 1. Right? If you recall in our discussion about the introduction to meiosis, I told you that meiosis has got two sequential cell divisions. Meiosis nanglola cell division ni tutu yungreswa cell chigne ni lisha ni di gogo hashire shilama chadur. So dilanga cell division ni do di nanglola. The cell division sequential cell division ni dilanga khala ore thangbo dila meiosis 1 che niwa dila meiosis 2 la or. So we have got meiosis 1 and meiosis 2. And meiosis 1 nangla and pro meta anatelo lan yoro. So dilanga khala prophase 1 metaphase 1 anaphase 1 telophase 1. Meiosis 2 le pro meta anatelo yore khare prophase 2 metaphase 2 anaphase 2 telophase 2. So in this video we are going to discuss about the prophase 1 of meiosis 1. Right, the prophase 1 of meiosis 1 nanglola roa. There are many important events that happens for the meiosis process. Meiosis process nanglo girwa ani event nenga chembo ne mangbo chunggo di ma songza ngajo gi thari video di nanglo la prophase 1 chekpo ngana jayenda. The prophase 1 nanglo la there are 5 subphases. 5 subphases ngayor. So it is a little bit complex, right? So but you have to pay attention. Okay, so meiosis 1 gi prophase 1 di. The prophase 1, right, simply called as prophase 1, is much longer and it is more complex compared to the prophase of mitosis. You see, mitosis ki prophase nyamdola compare chewe impa prophase 1 di ringa yore ro and nyamdola is complex higher even mangbo shuj chungor. I hope you remember what all happens during the prophase of mitosis. You have to do a revision of that one. Okay, so mitosis ki prophase le and prophase 1 di ringa da complicated higher resro and dinanglola subphases ngayor. My prophase 1 can be subdivided into 5 phases. Those 5 subphases are Leptotene, Zygotene, Pachytene, Diplotine, and Diakinesis. Right, so these are the 5 subphases of prophase 1. So you have to remember them in order. It happens in this particular order. Right, LZP, DD. Right, you can remember this sequence by saying Lalita Zara Pani De Do. So if you say that Lalita Zara Pani De Do, you know that L stands for leptotene, Z Zara la jomare dikhare Zygotene, Pani la di PA la di Pachytene, and De Do la jomare De la dikhare Diplotine, and Do la dikhare Diakinesis. So Lalita Zara Pani De Do. So if you say that, you can recollect the sequence of subphases of the prophase 1. So leptotene, zygotene, pachytene, diplotene, diakinesis, then you can resort to that particular phrase Lalita Zara Pani De Do. Okay, so diche. So also one by one to leptotene prophase 1 gi leptotene. Okay, so leptotene is also called as leptonema. Leptonema in Greek it means thin threads. Leptonema la the Greek is and thin threads. So why is it called thin threads? Because the chromatin materials they are just starting to become condensed, right? Chromatin material they are just starting to get supercoiled into chromosomes. The chromatin materials they appear like thin threads. So that is why the entire stage is called as leptotene or thin threads. Okay, so compaction of chromosome occurs throughout this particular phase. Leptotene chromosomes together condensed chaya go to work. Chromatin chromosome la chaya go to work. And chromosomes they gradually start to become visible under the light microscope. Since they are starting to become compact, right, chromosomes they gradually start to become visible under light microscope. Okay, leptotene and the nanglola harchesha, the centrioles or the centrosomes they have divided as well. Meiosis ki la interface yorda. Meiosis 1 di prophase 1 go mazunya interface yorda. DNA replication the sama chungsarda interface meomar the interface is very important. Interface chungsare DNA duplication chungsare chilo yogi cell organelle dige sama khare produce khare cheyagi material khare goyone dige sama synthesize chesare hakuya G1 S G2 phase la jo interface sama chungsar interface chungsa vejala ani khajir shine cell division chungya ko zura ani meiosis 1 gi prophase 1 leptotene la dire. The next phase khare leptotene ki jela khare la wimba na Zygotene. Prophase 1 gi Zygotene, second subphase. Zygotene is also called as Zygonema, which is in Greek means pair threads. Leptotene lawa imana kharimbe thin threads lagyo. Zygotene lawa imana pair threads. Pair thread thread ni. So why is it called as pair threads? Because there is a pairing of homologous chromosomes. There is a pairing of homologous chromosomes. Homologous chromosome dige khachi gore chati yung gor. Ta kina homologous chromosome na ha kugro. Chik pala jan le gyo na chikamla jan le gyo. Chromosome ki size ke chikpa khonzo kala allele ke chikba yo gyo dela ngazo khala gore homologous chromosome la gor. So there is a pairing of homologous chromosome which occurs during zygotene. So therefore it is called as paired thread stage or zygotene, right? So pairing of homologous chromosome is called as synapsis. So pairing of homologous synapsis lower, and synapsis is accompanied by the formation of synaptonemal complex between the homologous chromosome. Homologous chromosome ni parala synaptonemal complex sweve kengirwa ani kharcharu gore synapsis chungoro kuni nyamdo khajina shimba dagasme kuni nyamdo chadigo tenda mindoa homologous chromosome ni nyamdo dego dewa mindo kuni nyamdo khajin ligoro di homologous chromosome chikche di gare homologous chromosome ni nanglo ne chromosome chik di imbaina kuni nyamdo yongo jungzen khare lawe imbaina kuni ni parala synaptonemal complex form chigor. So synaptonemal complex lana khala ore de chromosome ni parala thik kauro no thongdo di khare dige sangma synaptonemal complex re. So di nenga chimburta homologous chromosome ni nyamdo yonga de nenga chimbure which happens during zygotene, right? Ani homologous chromosome ni nyamdo ledu zambala ani structure ji form chigda ta chromosome ni nyamdo leso dilanga jo khala ore the pair of synapsed homologous chromosome is called as bivalent or biledu zambala since there are two chromosomes, right? Homologous chromosome pair na yudu zambala mau da ngombo yudu zambala dilanga jo khala ore bivalent lower. And it is also called as a tetrad. Tetrad khajin lower tetrad nan shilo woro tetrad khajin lower lawembana it contains 4 sister chromatids. This sister chromatid chigre, this sister chromatid chigre. Wale sister chromatid chig nilan yoro. So there are 4 sister chromatids, right? 2 sister chromatids over here and 2 sister chromatids over there. Then therefore it is also called as a tetrad. So dikhare prophase I gi zygotene dire. So we have got zygotene over here. I hope you are clear regarding this one. And next dikhare lawe imbaina prophase I gi pachytene, right? Pachytene la dire. So pachytene in Greek it means, right, it means thick threads. Leptotene thin threads imba. Tene zygotene paired threads imba. Ine pachytene lana khala wore thick threads, right? Ani kola thick threads lago jumzen khala lago imbana the bivalents or the tetrads which started forming during the zygotene stage, now they become more visible because now they are starting to condense more and more. Okay, so they are becoming clearly visible, right? The bivalents they become more visible under light microscope, right? And there is a formation of recombination nodules. Now another important event over here, right? Recombination nodules form chigor tanji kunyi nyamdo dus jar deyo ta kharchi gore kunyi ni phazu tida thea gozu gor phazu tida thedu zambala ani kharchi gore there is formation of recombination nodules, right? Ta desia recombination nodule form chesa der da tanji kunyi nyamdo jar yo zygotene kabla kunyi nyamdo jar yo chromosome ni ta kharchi gore sister non sister chromatid ni parala tida thea gozu gor ti tida thea de langa zo khala gor lawe mana recombination lawor, right? It is also called as crossing over chungor tela. Crossing over occurs between non-sister chromatids, right, of the homologous chromosomes. Okay, di mao di da di ngombu di khare non-sister chromatids ra ngombu ni sister chromatid re mao ni sister chromatid ra ine mao do ngombu ni khare non-sister chromatids ra non-sister chromatid ni paral khachu gore thida theni crossing over chungor. This crossing over is very important because of this crossing over there is exchange of genetic material between the two homologous chromosomes. Kunyi ni parala genetic material phazu share chigor khajina ngombo di khesi pala jan imbaina mau de khachu jan le yor amla jan le yor pala gi jinte yor amla gi jinte yor ine dede khadu thegor la de jambala dele thegor so gene phazu dede khandes thegor la imbaina it occurs over here, right? Crossing over occurs between two non-sister chromatids of the homologous chromosome and it ensures the exchange of genetic material between two homologous chromosomes. Homologous chromosome ni parala genetic material phazu share chigor. And this particular process is called as recombination. Tadi kina sem na yagu nyama do desia. Kina remu dinalo siwe imbaina kuni ni pair cha ma se khachi do kuni phazu crossing over chungu do. Crossing over chungu zambala khachi do sister non sister chromatid ni parala genetic material share chidu zambala result desida. Result dikhare law imbaina dela gene khare chromosome digalo yogi material part chi khawachingsha amla gaji gi chromosome part chik di pala gaji chromosome gal phato do mau de leso e pala gaji gi chromosome part chashi chik khodo do amla gaji chromosome gal phato do so di khachi ra phazu de theso so this is called as recombination so the recombination la chik depending on the recombination khadu chungore la wemena it occurs during the Pachytene stage of meiosis 1 so the recombination la di khari drive chigore la wemena it's an enzyme mediated process enzyme guor tagas me kuni phazu share chigmaro tagas me chromosome thubgumaro it requires enzyme khari yorela na recombinase enzyme so recombinase enzyme helps in the recombination process. Recombination process nala kharchi gore non-sister chromatid ni parala homologous chromosome ni ki parala kharchi gore genetic material phazu share chi jugor. So enzyme recombinase process recombination and process recombination nala there is exchange of genetic material between homologous chromosomes. Okay. The next khare once Pachytene nanglola next khare la wimana crossing over results in the chromosomal recombination and by the end of Pachytene recombination is completed recombination chadur ine homologous chromosome ni loga chae marda homologous chromosome ni yamda rangda de de da kuni jani hajna synaptonemal complex de thando yorwa kuni ni parala de de the ni phazu thubre se da zere de ine kuni ni thando nyamdo de de synaptonemal complex thando yor and they are linked at the site of crossover yes chungsa jomare thanda kuni ni charsa jomare crossing over chungsa jorwa tela thande kuni jarni de der crossing over jar der recombination chungsa wi men thande kuni thu der tela wa so diche the next khare Pachytene gi je la khare la wi bana anju gi Diplotine so Diplotine lana khala or Greek word for since it contains di it means two threads Diplotine means two threads. The Diplotene nanglola, what happens is there is a dissolution or breakdown of synaptonemal complex. Thank you homologous chromosome ni nyam do jar jonge gi synaptonemal complex chomare protein denso yoro. Denso kharche do ore they start dissolving. Right? They start dissolving means the homologous chromosomes they become separated. Homologous chromosome ni dekhachi reta kuni ni separate che go zur kuni ni loga cha go zur. And they remain attached. They remain attached only at their crossing over site. Crossover chesama do shimba khajugore homologous chromosome ni the loga chart war except at the crossover site right the homologous chromosome they start to separate why because the synaptonemal complex they start dissolving adi kuni ni thusa de la kharchi do x shape do chag do dil x shape de la the x shape de la khala ora la women it is called as chiasmata x shape den zo la khala ire chiasmata la geta tik die themba nyama do chiasmata forms and Diplotene in vertebrates tadi das factoid childa in oocytes of the vertebrates the Diplotene stage can last for months or years tadi kana semna ya bunya ma do human reproduction nalo yang la girda human reproduction nalo la human females khonje gi subu nalo la egg meiosis la di ame khoba nalo no go zu war ma ji ne ame khoba khora nalo la kiran zu ovary nalo la oogenesis go zu sir egg go zu sir but meiosis complete chetu go mare egg produce chetu go mare khawala khar de gore la wemena it remains stuck at Diplotene Diplotene la khar de gore so vertebrates now la oocytes they can last Diplotene can last for months or years okay so this is Diplotene now the final phase is Diakinesis Prophase I the final phase dikha raha hai Diakinesis the fifth and the last phase. So now Diakinesis lana it is the Greek word for moving through something is moving okay so Diakinesis so Diakinesis na lo khar chungore la wimena thanji de la chiasmata la form che x shape de khar chi reta dus it is kisi cross shape do cha yo mena homologous chromosome ni loga cha do zambala chiasmata de ma tha ring du ro an kha chi re it will terminalize na pet ko end de le dir they will come to the end point of the chromosome telomere thi de wala le dur terminalization of chiasmata okay so during this particular phase the chromosomes they are now fully condensed chromosome de sangma bet ko to do ya pecha sa they are fully condensed ko chromosome shape ta da ko ya pecha chromatin ne chen gozu ne thread like structure ne chen gozu ne ta kharcha de ore rod like structure cha de chromosome cha de right and the meiotic spindles the spindle fibers jo mare so microtubules they start to assemble and prepare the homologous chromosomes for separation ta homologous chromosome ni de phazu separate shela taji homologous chromosome ni de nyam do synaptonemal complex ki jar dewa 아니 딩에기 디플로틴 페이스 나로 코니니 브레이크다운 치니 로가 차야 고조엠바 아니 사이납스 데라 자르데요 so synapse de tat chela ma khachi go gore synapse de meo syo go gore ro synapse de meo syo go du zambala ro ani khachi gore the microtubules spindle fiber ki microtubule den so assemble cheni ro kuni ni logo syo go zu gore so 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 ani loga chago marda tanda loga chagi urda okay so meiotic spindles is assembled to prepare the homologous chromosomes for separation and by the end of diakinesis diakinesis zogal hamula ani khachi gore nucleolus la ro nada nucleus na lo ge ro nucleolus chomarwe ko disappear chadu gore nuclear envelope starts to break down nuclear envelope it starts to dissolve it starts to break down and diakinesis it acts as a transition into the next phase that is metaphase 1 the prophase 1 gi jela khare metaphase 1 ro so diakinesis ki the end part chomarwe ti khare it acts like a transition into the metaphase right before which what occurs nucleolus la dure nuclear membrane ge la dure and spindle fiber dige sama khachi re homologous chromosome ni la pha jare go chu ro and they start to pull each other okay so that is about meiosis 1 meiosis 1 ki prophase 1 dirda so ngazu meiosis 1 ki prophase 1 na lo la ngazu khar karta re leptotene zygotene pachytene diplotene and diakinesis so I want you to write down the notes and write down the diagram for each and you need to remember what all happens during each of these subphases of prophase 1 very important pe nenga chimburta so ma kena zo reproduction chapter na jang du zamba ge nenga chimbu cha dur so evolution jang du zamba ge nenga chimbu cha dur so then zamba you have to remember what all happens during leptotene zygotene pachytene diplotene and diakinesis so you can practice drawing the diagrams and after drawing the diagrams you can write down notes for each of these diagrams diagram lata ni kena zo deshe kya yor so okay so in the next video we are going to discuss about the metaphase 1 anaphase 1 telophase 1 and prophase 2 metaphase 2 anaphase 3 chela ngaju remaining part thing also j video na tayanda okay so please read about meiosis and you can watch the next video okay thank you